What are some surprising findings or unexpected obstacles that you've run into while doing, let's say, the biohybrid electrodes?
I mean, biology is just, when it works, it can do a lot of things that humanity is just not at that level of capability yet. But also in neural engineering, or either, whether that means systems neuroscience or BCI, you'll start in mice, and then maybe you'll work in an intermediate species like pigs, and then eventually end up in monkeys, then end up in humans. And when you have an electrode, or even something like optogenetics, that works basically the same in mice as it does in monkeys as it does in humans. When you're engrafting neurons into the brain, I mean, there's a big difference between mouse neurons and human neurons, and macaque neurons are a different thing entirely. And so you end up having to redo a bunch of this work in each species that you work in. And so every time we switch species, there's a lot to relearn. And 15 years ago now, probably something like that, there was a major discovery of the ability to turn any cell into a stem cell. Again, this was a discovery called induced pluripotency, won the Nobel Prize a while ago. And that works really well in rodents. It works really well in human cells. Turning a macaque skin cell into an iPSC is like, there's just a bunch of little tricks that don't work as well. And so the biology is pretty deep in a lot of these areas.